There we go. So as I said, I'm going to go back to chapter 9, and I'm going to start, start once again kind of going over that and drawing these parallels between these two different kinds of processes. And the reason why I want you to do that is, like I said, one of those highest ways of thinking, and one of the things we try to teach you in school, and maybe I've said this before, but I bet not about this particular subject, but whatever class you're in, we try to teach you how to think. And you think, what do you mean how to think? Well, how to process information and how to understand the necessity for reliable information. You know, it's okay on Wikipedia, but remember, anybody can write on Wikipedia. So check your sources, learn how to communicate. You know, you understand those things. We've been trying to, well, education systems have been trying to teach you that since you were in the first grade. So there's no surprise in all of that. So that's what I'm going to try to do today. I'm going to finish up the photosynthesis chapter. Let me get ahead to where I was. And then I'm going to go back to the respiration, and we're really going to do that compare and contrast thing again, because they are very similar. And the danger to me, or the danger to you, and the danger I've seen to you guys with this concept is you'll get them all mixed up together. You'll get them all screwed up in your head. And then when I ask you them on the exam, you'll tell me about photosynthesis when it means respiration and the opposite. Okay? And you can lose a boatload of points on those kinds of questions, especially if this is what I call a systematic error. Systematic error means you're probably not guessing. You really have it backwards. If you get all the ones that are in the dark reactions wrong, you say it's the light reactions, and all the ones in the light reactions, you tell me, no, that happens in the dark reactions. Well, at least you're consistent, but you're consistently wrong. What would you call that in science? The difference between accuracy and precision. Precision is getting the right answer each time. Accuracy is getting the same answer each time, right? Kind of a philosophical thing. You could be accurate, but not precise. I mean, you could say 2 + 2 = 5 a million times, and I guess you'd be accurate. Okay, all right. Calvin cycle. Let's focus our attention on what we need to know out of these things. Let's not waste our brain cells. It's always good to go through the cycle so that the names like 3-phosphoglycerate, 1,3-bisphosphoglycerate, the author was kind to us, at least in this picture, and gave us G3P, which is glyceraldehyde 3-phosphate. I'm not asking you to know those names. I do want you to understand what's going on, right? So like carbon dioxide from the top, and all throughout the book, they've tried to stay consistent with coloring, that purple things are proteins. If you remember back even in the early chapters when we talked about proteins, they were most often, if not every time, they were proteins. Even the photosystem we looked at the other day, remember, it was purple. It's a protein, okay? So in this case, proteins, which do all kinds of weird things, this is an enzyme, and the enzyme takes a couple of substrates. Enzymes can take 1 substrate, 3 substrates, 5 substrates. The whole point of the enzyme is that something goes in and something comes out. So in this case, here we go. This is what's coming in, right? And of course, you look at that and you realize, once again, like we talked in class, this is that whole beauty of Mother Nature, understanding the necessity for continuance, for resilience, for the ability to keep going on, as long as you add a few things to it. Build a system, a factory, and just kind of add a plant, or the electrical generating plant isn't going to work. The pistons, or however those things were, the generators, and I kind of know how they work. But anyway, they're not going to work unless you put in gas or coal or something like that. So you need fuel. You're not going to make cars in a car factory if you don't have people making the cars and things to make the cars with, the metal and the rubber and whatever else you put in a car, right? Same thing here, isn't it? The Calvin cycle, the Calvin-Benson cycle, the dark reactions, okay? Make yourself those lists, like that thing I showed you in the slide. Oh gosh, let me pull up my other computer. What's the figure? 1022b. All right, so here we are like I said, in the dark reactions. And the dark reactions is a factory, but a factory needs energy, yeah, right there, and it needs a source of materials. Okay, so, beyond the chemistry, beyond the words, reduction, oxidation, phosphorylation, dephosphorylation, understand what that means, understand what that does. Okay, ATP, when it's converted into ATP up there, ADP up above, right here, that first one, that first part of the cycle that turns that 3-phosphoglycerate into that other stuff there, that requires energy. What you're witnessing there, and I can link a lot of words together here too, folks. What you're witnessing there is hydrolysis, right? You're witnessing that process by which a big organic molecule, remember that way back in, well, I don't know, chapter 3, 4, 5, I think it was 5. We talked about polymers and monomers. Okay, well, look at this. ATP, 3-phosphate, see my fingers? ATP is 3 phosphates, bigger molecule. ADP is 2 phosphates, smaller molecule. Okay, so we turn a polymer into a monomer. When you take bigger things and make smaller things, you not only make them smaller, you also release energy. It's exergonic. It's exergonic. So the ATP going to ADP is exergonic. Whoa, where'd that energy go? Energy can't be destroyed, the laws of thermodynamics say. It's got to go somewhere. It can only be converted. So where did the energy go from the dephosphorylation? Now we're getting some jargon, right? Where did it go from that ATP being broken up? That's what turned the cycle. That's what, took the chassis of the car and then put the frame on it. That's what turned 3-phosphoglycerate into the other stuff, okay? Remember we talked about that. The big part of that first phase that's going on is something called carbon fixation. The carbon dioxide is atmospheric. It's Rubisco's job as an enzyme to take the RUBP. I ain't going to bother to remind you what that stands for. Don't worry about it. Take that substrate, take this substrate, carbon dioxide from the air, and combine them and make the stuff to start the cycle. Now you have a little energy, you go a little further. In phase 2, if you're wondering what these phases are about, right? Carbon fixation, to grab carbon, to fix it into a chemical compound, to turn it from CO2 into this stuff. See, the little gray balls are supposed to be carbon atoms, okay? Phase 2, you reduce. It's like a cookbook thing. You need ingredients. Phase 2, you're going to reduce. You take the 1,3-phosphoglycerate. I'm trying to do a French accent, but I'm not doing it very well, so sorry. And you reduce it. You reduce it. Now, what else happens is the phosphates are disconnected. See, I told you the ATP becomes ADP. The phosphates are disconnected. Now, again, the 6s are all the balancing, so you can ignore the numbers right now. But I didn't say that those phosphates, when they're chopped off of ATP, immediately go to ADP, okay, or just gone. Sometimes what they do is they attach themselves to things. Like, for example, if you look, that's where they went. Now the energy was released, but that phosphate that was chopped off was actually, she was attached to something. All right, whatever that something, you reduce it. So you put electrons on it, reduce it, put electrons on things, reducing. Now is reducing an exergonic reaction or an endergonic reaction? When you reduce something, do you build something up or do you tear it down? Think back to what I just said, though, about when you are reducing something, you're giving something electrons. Are you tearing it down or building it up? If I reduced you, I would be giving you an electron. So what did I do to you? It's an endergonic reaction. That's right. When you are reduced, you're building something up. You're taking small to big in a real general sense, right? Smaller molecules. Is that my mower? Oh, geez. Smaller molecules, and you're making larger, more complex molecules. My great-great-uncle lived in Texas underneath shelter half that he had on the side of the river in Texas. I mean, when they first moved out there, this is like a long time ago. There's a picture of him standing there. I asked my dad, I said, what's that thing that's in the riverbank there? And he goes, what do you mean? He goes, that's his house. And I said, that's not his house. That's a part of, shelter half is like half a tent. So it's just a tarp that he had put into the sides and the cliff and then put down into the riverbed. And I'm like, what? So then there was another picture of my grandmother having, he's standing in front of the tarp with him. I said, who's saying? Well, that's his wedding day. I'm like, oh my God. So here, honey, climb under the tent with me, the half a tent. If you want to turn that tent into a house, you're gonna have to get some things. You need some wood, you need some flooring, you need some plumbing. Well, maybe they even have plumbing back then. You understand? That's what reduction is. We're building a molecule up. Okay, we're making a complex molecule. That's because we're overall, we're taking a really simple molecule, CO2. Get rid of my scribblings. And we're turning it into, down at the bottom, glucose and other organic compounds. Food. Okay, food. That's what plants do. They photosynthesize. They make food. Now, as I said in class, Mother Nature is really, really clever. So the last phase is probably the beauty of this factory. This factory never runs out of machines to do the tools. So again, it is like a car factory. The car factory could sit empty, but the tools that are there, the thingamabobs you need to make a car, which, anyway, they would still be there. The tire making machines, the bumper making machines, and all that kind of stuff. You know, all that stuff would still be there. Now, yes, I know it decomposes and so on and so forth. And so would this, if you didn't have energy, right? This cycle, this Calvin cycle would do the same thing. But at the end, you turn the stuff you need to start with, right? You turn RUBP, or you create RUBP from 5 of those molecules that he eventually turned into food, right? She's not greedy, Mother Nature. She says, I could use all 6, but listen, it's actually going to be more beneficial, more efficient. No altruism among, you know, I'll save you their molecule, but more efficient chemically. If only 1 of those is turned into sugar and 5 are left, it's this whole process is exergonic. You can't drive a factory without energy. So to get it back up to RUBP where you need to start, you have to put in some more ATP. Okay, so ATP is the energy, NADPH there on the right is the source of materials, source of electrons, and overall you're cranking out sugar at the bottom. Think of it like a faucet down there, right? And you're doing that by using that energy and using those materials. And as long as there's carbon dioxide, plenty of that around, as long as there's carbon dioxide, you'll be able to turn it into something fancier. You'll take CO2 and make C6H12O6, right? Glucose. You can't live on carbon dioxide, but you will if you're able to do this. Now, the glucose has energy. That's why you can use it in the previous chapter in respiration. And where's that energy? Well, there are plenty of indications right here where it is. There's the ATP there on the right. There's the ATP on the left. Exergonic reaction or endergonic reactions when you do the reduction in phase 2. Plenty of places where energy is being transferred. And if you remember, I don't know why I did that with my hands. And if you remember, all of this stuff that's coming out of here, let me get rid of my scribbles. Wish it would default to, anyway. All this stuff goes back to the light reactions. So if you're going, okay, let's trace the energy back, the energy in food, right here, I can point to it. That's where you're getting it. And then where's the energy coming from there? Well, in the light reactions, and in the light reactions, this thing is giving them energy. Bob. You guys know what Bob is? Bob. Military slang. So if you're in the military, you might know what it means. When you look up in the sky and it's hot, what do you see? And it's clear day. Not directly at it, I wouldn't do that. Yep, so BOB is big orange ball. My dad used to say that sometimes in the military it's important to make it as simple as you possibly can so that there's no misunderstanding about big groups of people. Big orange ball. All right, so you understand what's going on there, right? All that sunlight energy is being trickled down, slowly released. Don't want to do it all at once. That's what the shuttle does, right? We get that rapid release of energy. We don't want to do it that way. We're just kind of sucking the energy out of those electrons. That, you know, back in the photosystem, there was that magic. It's kind of what I used to think about photosynthesis when I was in college. Magic. There's that magic, if you will, but not really magic, where the sunlight energy got incorporated into an electron. An electron's energy went up because it absorbed sunlight energy. You know, nothing important, but just the basis of life, okay? Just the whole basis of taking something that you can't live on, carbon dioxide, and making food out of it. All right, we got to move on, okay? You understand, of course, the difference in numbers. For 1 revolution, there's going to be 9 ATPs. and what 6 NAD pHs are going to be used. So there's a disparity there. There's a disequilibrium. There's a definite possibility. There's a certainty. There's a certainty that you're going to run out of ATP first because you're using it faster. So that's what the purpose of this was. And I want to go into this right now because I want to kind of review the whole electron being transported thing. But remember, this is on the thylakoid membrane, and this is how we're getting that ATP that we need. Remember, ATP is ATP anywhere, but in this case, we have a really special role for it. We have a place for it to go already. So I'll come back to that in a second because I want to get to the new stuff today and then go back to this. So, am I at the right place? Yes. Okay, this is the same picture that we looked at earlier on. Is it? Not exactly the same, but the one we looked at earlier on the first few slides. So, because what I'm doing is I'm shifting my attention, but I did mention it. I mentioned these things on the bottom, the stomata. Okay, so what the stomata are is, let's see how well I can draw this morning. I remember not to lean to the screen because you guys are always having to look up my nose. Not conducive to learning, I would think. All right, so guard cells, which are like, let's say, beans, if you will, right? They're touching each other. And this is a hole, the stomata. The dashed lines indicate it's underneath the guard cells, right? Guard, G-U-A-R-D, guard cell, just like you have somebody at a fence. So if these guard cells were to swell, they would cover the hole. And then I can't erase it. No, I can't erase the whole. I erased the whole thing. But then when those guard cells would shrink, okay, then they would open the hole. So that could be very easily triggered. That's nothing fancy. That's just a couple of cells getting bigger. Now, you're just talking about millimeters, but we're talking about letting gases in and out, carbon dioxide and oxygen, and they're not very big. So plants essentially breathe, kind of in, well, they do respire, of course, but kind of in the reverse of us, they do photosynthesis. So let's kind of focus on this whole stomata, carbon dioxide, and the problems that plants have with their stomata. You see, Mother Nature, like everybody in my bio too, the thing I try to get across is that evolution is not perfect. That if it was perfect, you'd have to go back to the very beginning and do all the right choices. Evolution is kind of like making the most out of what you have right now. And even if you could go back to the beginning and make all the right choices, Mother Nature changes everything. She changes the environment along the way. 250 million years of ruling the world as the dinosaurs. One day took about a decade, I think, about 10 years after the asteroid strike for all the dinosaurs to die off. I don't know if I have that number exactly right. It didn't take very long. All right, so here's what plants are going to be doing to try to make sure that they can survive, because what's going on is the environment on Earth is changing and plants are finding different places to grow. And it's always kind of nice to find a new place to grow because maybe you don't have to compete with other things to grow. All right, so here's what the problem is. CO2 and carbon dioxide and oxygen are exchanged through the stomata. We've said that a couple of times. There is another problem, though, or there is a problem. When you open your stomata, especially in an environment in which we call arid, I'm going to write the word. Oops, I didn't mean to be in caps. Arid. Arid, like the underarm stuff, right? Arid means dry. I think the underarm stuff has two eyes, by the way. The desert is arid. And then there are regions well, there's everything in between, obviously. There's the tropical rainforest. That's the opposite of arid. But then there are regions that we might call, in sort of a loose term, semi-arid. Semi-arid regions would be like, for example, the Midwest, United States, the breadbasket. You know, where it's, you know, it rains, but, you know, there are times in which it doesn't rain for a while. Farmers have to suffer through that and all that kind of stuff, right? There are droughts that occasionally occur out there. But I guess I would say it, luckily for us anyway, unless global climate change has something to say about it, we're still growing food, a lot of it in the right place, that place where you get in, it's Goldilocks, just enough rain, but not too much. And there's everything in between semi-arid and arid. So plants have a problem in these drier climates. The drier it gets, the more of this, I'm writing it on the screen, the more of this is lost is when you open your stomata, or the more of this transfers. Okay, so there's more water inside. It's all diffusion. It's all diffusion. No magic here. It's not osmosis because there's not a membrane. There's an opening, a hole, a stomata. There's more water inside, okay, of the plant than there is on the outside. It's relative. I know that photosynthesis uses water. Of course it does. We talked about that in the light reactions, but there ain't much or hardly any outside. So diffusion, no matter how little each of you have, there's always in diffusion to work, there's a gradient. So the water is going to flow outside. The plant is going to dehydrate, just like you would do, just like you do when you sweat. Not very effectively in Florida, by the way, because the water never evaporates. It never takes away the heat. That's always the problem. So let's address this issue of water. What's the big thing about water? Well, you can't dehydrate, obviously. Although resurrection ferns have figured out an interesting way to do it and come back to life, actually. They completely can lose, I think, almost all of their water in their cells, and next time the rain happens, that's why they're called resurrection ferns. The ones that grow out of the trees. Scientists are like, how do they do that? All right, so you can't do that. You can't dehydrate. So you can close your stomata. All right, simple enough. Close your stomata. You don't dehydrate. Yeah, it's like holding your breath. Yeah, it's like holding your breath because now you can't bring in any carbon dioxide. So you can't do photosynthesis because remember in the Calvin cycle, you got to have carbon dioxide. So that's a problem. Okay, well, you got food stored up, or you can live for a while. And there's another problem. Not only are you not able to make any food in terms of not bringing in carbon dioxide, there's a problem with the oxygen. Okay, now here, let me show you what it is chemically. So here is the Calvin cycle we just looked at. The very same one that we went through, just a few minutes ago. So once again, here's the carbon dioxide. So nothing new on this screen, nothing new at all, because I want you to focus your attention on what I just circled, the carbon dioxide at the top. All right, imagine for a second, here's what's going on. The stomata are closed, right? Not losing water, it's dry outside, Paul, close the stomata. So we're not losing water, so we're not dehydrating. Good, that's good, check, plus, right now. We're not bringing in carbon dioxide. Okay, all right. The problem, of course, once again, is how long that goes on. Now, chemically, other than not bringing in the carbon dioxide you do have, maybe have some stored up, because like when you hold your breath, you got a little bit of oxygen in your lungs for a certain period of time. Some of those folks can dive down to, 400 feet and hold their breath? Okay, but the little bit of oxygen you do have starts jumping ship. It's like rats deserting a sinking ship. What do I mean by that? All right. Enzymes have an affinity for things. Affinity, they have, they fit. Puzzle pieces have an affinity. Your key in your car has an affinity. You get it? You might have an affinity for a friend of yours too. It can be things fit together, or you feel a connection with your friend or loved one or anything like that. You might have an affinity with your dog. I think I have an affinity with Cooper. Right, Cooper? Oh, he's asleep, so. Okay, so anyway, enzymes have an affinity, but unfortunately, you get a couple of marriages under your belt. Hopefully you won't, or if you have, you realize, of course, that affinities go away. That sometimes, if I took a file to my key and I ground it all down, it would no longer have an affinity for the lock of my car. That would be a stupid thing to do. Cooper could get rabies, and he would no longer have an affinity for me. Hope that doesn't happen, knock on wood. You could grow to dislike somebody, and you would no longer have an affinity. Well, guess what? Rubisco, the enzyme, with its active site, especially when it gets older, especially when they're older, that's going to be more pronounced because of entropy, things falling apart. Its affinity for carbon dioxide can be affected by oxygen. It actually can be affected by oxygen completely, you see, because when there is a choice for Rubisco, when there is a choice between oxygen and carbon dioxide, it sometimes will switch over to oxygen. Okay, now the problem with that is, chemically speaking, when you switch over to oxygen, so I've replaced the CO2 with the oxygen at the top, you get nothing. Nothing happens. The cycle doesn't turn. You may have the ATP from the light reactions. You may have the NADPH. You're going to have a lot of RUBP sitting around, but the enzyme there, Rubisco, unfortunately, this is the first part of the factory. The switch is off. It's not going to turn. Now, this process, so I can give you the official name, is known as photo because it involves light, right? It's sunlight again. And since it involves oxygen, not because it involves the process, but since it involves oxygen, it's called photorespiration. When Rubisco switches over to start a binding to oxygen, Now, is this a good thing? No, 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 no. This is not a good thing at all. This is a bad thing. This is a very bad thing. Plants, when they do this, when they photorespire, make no food. Okay, so it's okay for a little while, but you can't do that very long. Now, if that drought lasts for a while, okay, if that drought lasts for a while, or even if you don't get a drought, but during those really peak warm times of the day, and those of you that may have grew up in the Midwest know in the middle of the summer, I didn't grow up in the Midwest, but I do know in the middle of the summer out there in the fields, it can get pretty doggone hot, okay? It can even get pretty hot and even dry without being in a drought per se, right? Just in the heat of the day. Okay, so during those times in the heat of the day, these plants that grow in these regions, what they're going to do is they're going to start closing their stomata. And when they close their stomata, they have some CO2, but they can't bring in any more. That's a problem. But more importantly, the oxygen can't be released through the stomata. Remember, that does two things. It brings in the CO2 and lets out the oxygen. Of course, that's the oxygen goes off into the air, and we breathe. And plants do too, because they respire, right? So none of this happens here in photorespiration. Now, how do plants deal with this? Well, they hold their breath, essentially. They kind of wait, and they hope that these dry conditions stop, that it starts to rain again. They can open their stomata, not worry about losing water, and start bringing carbon dioxide, and more importantly, like I said, getting rid of that oxygen. You see, Rubisco, as I said, will bind to carbon dioxide and oxygen. But if you remember back in our enzyme chapter, when I talked about competitive and non-competitive inhibition, even when I talked about feedback inhibition, is about numbers, right? If there were, and let's just really make this outrageous, 1 million oxygen atoms and 10 carbon dioxide molecules, if there were 1 million oxygen molecules, because they occur O2, and 10 carbon dioxide ones, do the probability. You might bind to a carbon dioxide every once in a while, 10 out of, what did I say, 1 million? But the probability is you're not. So if you do that bad probability, if you don't bind to carbon dioxide enough times, which you're probably gonna during photorespiration, you're going to die. Simple as that. Now, many plants are on Earth are what we call C3 plants. C3. Don't get real fixated on it. It actually is a way that we can differentiate them from these other types of plants I'm going to talk about. And the name comes from, let me go back a slide. The name comes from the fact that predominantly, or a lot, what you see during this whole cycle are 3 carbon compounds. I'm circling them. 3 carbon atoms. Now, it not all the place, right? But predominantly what we're seeing is a 3 carbon atom. That's where its name comes from. C3 just helps botanists know which cycle we're talking about. Because living things, in order to survive in those climates, they can't do anything about dehydration, you see. The problem with dehydration is it's diffusion. It's natural. It's normal. It's falling off a cliff. There's nothing you can do about it. But what organisms do, if they want to survive, most times they don't. They go extinct. Talk to my Bio2 students. But if they want to survive, what they do is they find a way to deal with it. Okay, so this is where we're going to go. This is a reality, photorespiration. Now, if you live out in the Midwest, from a farmer point of view, right, from someone who grows crops, makes their money off of this stuff, you would like to limit, or gosh, if you could, stop photorespiration. Even when it's not a drought, and probably the biggest concern, or one of the bigger concerns in drought, is the actual drying out, the plant dehydrating regardless, right? If your stomata are closed and there's still no water and your roots have nothing to suck up, you're gonna die of dehydration. But even in the heat of the day, they probably would like it if they didn't undergo photorespiration, because they wouldn't lose money. Because every time it doesn't make food, every time it doesn't make a glucose, it's a factory, folks. It doesn't grow. And if it doesn't grow, your wheat doesn't get taller. You can understand that from an agricultural point of view, they're very interested in this whole thing of photorespiration. Now, is there a solution? Well, there is. We haven't necessarily come up with this solution. Or at least initially. I say that not knowing exactly if there is a chemical way we figured it out, but Mother Nature has. And let me show you how clever she is. There's another type of plant known as a C4 plant. Again, don't get hung up on this, but a C4 plant is because right here, I'll talk about this in a second, it makes a 4-carbon compound. Let me close the window because I hear the mower guy. All right, so it does make the 3-carbon compounds too, because down here is the Calvin cycle. And look, here's another 3-carbon compound you might remember, pyruvate, back from respiration, same molecule. So the Calvin cycle is only C3, and this cycle here is the C3 of the Calvin cycle and those C4s up above. Again, just to try to keep them apart. All right, now here's what we say. These plants have figured out a way to get to deal with this photorespiration problem, which I'll explain in just a minute, by doing steps in different cells. You see, if you cut open a C4 plant, you would find, first of all, the vein. That's that's like the blood veins of the cell. The pen just died. Anyway, that's like the arteries and the veins of the cell. That would be the vein there. And then you got the bundle sheath cells, and they wrap around the vein. There we go. Right, these right here. And then around those are a bunch of sort of scattered cells known as mesophyll cells. And then on the other side, this is the epidermis. So this is looking through a leaf. And like I said, the stomata are on the bottom. Well, that's a good question. Why are the stomata on the bottom, do you think, versus the top? Why on the bottom of the leaf versus the top of the leaf? Question of the hour. Give extra credit if you guys answer questions. I just worry that actually gives an advantage to someone who just has an abnormally fast internet connection. But, well, they don't really want to do that, right, Gavin? They don't really want to release the water. But which side of the leaf of a plant would you think gets more sun and therefore subjected to more heat and dehydration? Sure. So you put the stomata on the bottom, okay? All right, so here's how these C4 plants deal with this photorespiration. Well, they really don't, because it still can occur, all right? So what they're doing is minimizing it. Your stomata are going to close. The stomata, they're going to close. They're going to close because you don't want to dry out. It's like, if the ship is sinking or on fire, you probably ought to stop it from sinking first, okay? So here's the same sort of mentality. The reason why the cells are arranged is because that's important. In C3 cells, the cells are just kind of scattered in amongst each other. They don't make what they call them bundle sheath cells. They don't make this sheath, this wrapping around the vein. All right, and they have the mesophyll cells. So again, what we got is 2 places, 2 different cells. Now over on the right is the picture with the chemical, what the heck is going on, illustrated. There's the mesophyll cells. There's the bundle sheath cells. And the idea of the vascular tissue is, of course, that's where the food's going to go, because you can move it throughout the plant. All right, what's going on here? Well, here's carbon dioxide. Now, carbon dioxide has been brought in through the stomata, just like normally, but then the stomata close. And so the carbon dioxide levels drop, and there's nothing you can do about that. Maybe you can wait until it, starts raining again. I mean, in the end, if it stays dry long enough, ain't nothing you can do. You're just going to die, okay? This is one of those things to try to mitigate it. So the reason why you can survive when the stomata are closed, these C4 plants, is because of another enzyme. Look at it right here. It's even named correctly. PEP, which stands, I'm sure, for some acronym, but carboxylase. It's an enzyme. Now, guess what? PEP carboxylase is like Rubisco. Notice what it's doing, right? It's grabbing, it's fixing carbon dioxide. See that? It's turning carbon dioxide. That enzyme carboxylase is turning carbon dioxide into this thing, one of those 4-carbon compounds, oxalic acetate. Again, let's not worry about any of those, okay? So you got this enzyme that's taking that carbon dioxide and it's fixing it. Now, the difference between it and Rubisco is it doesn't have the same affinity problems. In other words, it doesn't like oxygen. It doesn't bind to oxygen. It only binds to carbon dioxide. So what they've done, or what the C4 plants, the C4 plants, they're all behind everything. What they did is they built another enzyme. Another enzyme was constructed that did not have these problems with switching. Is it carbon dioxide or oxygen? There's a heck of a lot of oxygen around, right? No, it doesn't like oxygen. It always takes so you're going to suck every last carbon dioxide atom out, before you get to a point where, the stomata can't open again, okay? And you run it through a series of reactions, oxaloacetate, malate, and this is all just chemically determined. This is why it has to happen this way, okay? And then you can see the little opening. They're probably plasmodesmata. They're probably those things we talked about way back in chapter 6. Through those plasmodesmata, malate is pumped. And the reason why it's pumped is because simple diffusion. There's more out here of malate than there is in here. Okay, so the malate is going to diffuse across the stomata, so it's going to be constantly pumped into the bundle sheath cells. Now, what happens in the bundle sheath cells is the carbon dioxide that was fixed before by PEP carboxylase is, well, essentially, it's kind of unfixed. It's released. Okay? So then that can run through the Calvin cycle and everything's just fine. Everything's just hunky-dory. The pyruvate that I showed you about earlier, that's what comes out of that. Pyruvate is when carbon dioxide gets incorporated in the Calvin cycle. When malate releases carbon dioxide and pyruvate, or carbon dioxide, it also releases pyruvate. All right, now, this does work. This is effective. There are plenty of C4 plants around today. They typically live in these semi-arid regions, these C4 plants. A lot of times C4 plants are things like sugar cane, and many times agricultural crops, which again is important to farmers. So this does work, but it does come as a price. Now, you remember in the Calvin cycle, there was no instance where, in the respiration cycle, other than glycolysis, you didn't use ATP. In other words, throughout respiration, the net yield of ATP was always in the positive. Even if you only did glycolysis, you got +2. And then if you did the whole thing, you got 32, 34, 36, depending, right? Here you can see that in this whole process, there is a price to pay, though. You do need to use some ATP. So while you're making food down here, in other words, using energy to do something constructive, to make food, you also need to spend a little bit of energy to keep the cycle going. Now, obviously, you get more down here than you get up here, right? Or it wouldn't be efficient. Mother Nature wouldn't have evolved it that way. And what you do when you add the energy is because that's necessary. Guess what? Here's another one of the cycles. Isn't that cool? To keep moving PEP around. That's what you're doing that with. You take the PEP, the CO2, you take the PEP, you take the CO2, you take PEP carboxylase, the enzyme, and you make oxaloacetate. So you see what I mean by it saying it solved the photorespiration problem, because what you got going on down here in the pyruvate, let me erase my scribbling, is even though the oxygen levels might be rising, for one thing, oxygen finds it more difficult to diffuse across, this way. Okay, and even though the oxygen levels might be rising to the point where they just start squeezing through, there's nothing you can do about it. You're always hoping, at least at the beginning, till it's too late for everybody, you're always hoping that there's more CO2 than there is oxygen. As long as it's a million, well, to make it really ridiculous, 1,000,000 to 1,000,001, you still got a preference. Obviously, it wouldn't be numbers as silly as that. Okay, so it becomes a relative concentration thing, just like competitive and non-competitive inhibition. Which one is the enzyme going to bind to? The inhibitor, or there are two types of inhibitors, or the substrate. Which one are you going to take? The one there's more of. It's an enzyme. It's a molecule. I can't think. Now, another reason why this is beneficial is the light reactions. You remember in the light reactions, one of the things that comes out of the light reactions is O2. Remember, the light reactions, water gets split, right? Because all throughout photosynthesis, we're following the electrons and the protons on the hydrogens, right? The hydrogen atom is made up of just one proton and one electron, way back to chapter 2. So what we've been doing is energizing that electron, giving it energy, moving it to higher energy shells, going back to chapter 2, and taking that proton and pumping it across pumps, letting it flow back through the ATP synthase to make ATP. Whether we did it in respiration or we did it in the light reactions, we did the same thing. Now, those are happening out in the mesophyll cells. So that's really where the oxygen levels are rising, because you're splitting water. Remember, what plants do is they take everything off of water but the oxygen. So while the oxygen levels are rising, because again, your stomata are closed, there's nothing you can do about that. You're not bringing in CO2. You're not getting rid of oxygen. So out there in the mesophyll cell, there's really a problem with oxygen rising. It's rising, it's rising, it's rising, but guess what? PEP carboxylase don't care. PEP carboxylase says, I don't like oxygen. I only like CO2. Remember, Rubisco's different. If the oxygen levels got higher and higher and higher down in the bundle sheath cells, Rubisco would start going, man, gosh, I really kind of like CO2 too, or I like oxygen too. So, but unfortunately, every time it binds to oxygen, now does it stay binded forever? No, just like the inhibitors don't. Think of oxygen in that case, there you go, as a competitive, it might be non-competitive, I don't know, it's probably competitive. Just think of it as an inhibitor. Oxygen is an inhibitor, right? Oxygen is an inhibitor to the Rubisco enzyme. Okay, so that's how C4 plants do it. If you think that life just comes up with one answer to things, oh no, you're wrong. You're wrong. There's a million different ways to so-called, sorry to all the kitty lovers out there, skin a cat, right? There's a million ways to do things. It's all just chemistry. You know, NASA defines life as this really complex chemical dance. So there's other plants that have figured out to do it differently. And these are known as CAM, C-A-M, plants. If you're really dying to know what that stands for, because you know anything about science, everything's acronyms, it stands for this. Racemacin acid metabolism. Now, if you're wondering, well, where in the heck did they get that name? It's kind of like the C3 and the C4. Somewhere in this reaction that you see on the right, this is the CAM plants. The one on the left is the one we just went over, okay? The one on the right is the CAM plants. Somewhere in those sets of reactions, crassulacean acid is made. It's probably the organic acid they're talking about there. I haven't really examined the crassulacean acid metabolism specifically. So what do I want you to know about it? Well, here's what I want you to know about it. This is another answer. Pineapples, for example, are CAM plants. Cacti are CAM plants, ones that really deal with dry climates too. What I want you to know about it is, unlike in the last ones, in the C4 plants, where the answer was to separate the steps into two different cells. Remember, up here you had one set of reactions in the mesophyll cell, and remember that's also where the light reactions were happening. Only the dark reactions are occurring down in the bundle sheath cells, right? And that's in a different spot. So what you did is you separated the steps. You separated Rubisco from its inhibitor, oxygen, by space. That's what spatial means. Spatial means space. Two different places. The oxygen levels are going to rise in here. And again, they might spill over, but they're going to rise in there a lot faster than they ever rise here. So you're hoping that whatever little oxygen or carbon dioxide you do have, that you're pumping constantly into that bundle sheath cell, is always greater than the O2. So on the left, let's do a little chart. On the left, you hope that the CO2 concentration always stays higher than the oxygen. Right, that's what's going on in that cell down below. PEP carboxylase doesn't have that problem, doesn't like CO2. Okay, CAM plants do it another way. They do it by what we call temporal, which means time. They do it by temporal separation. So here's what they mean. They open their stomata at night. This is a lot simpler too, by the way. They open their stomata at night, right? Even at night in the dry regions, it's not necessarily as dry. Remember, it's all just got to be about a little bit. Living things just need a little bit to survive. As long as you're getting more water than you're releasing, even if it's, and it's never going to be, well, maybe it is. It's never going to be as small as one molecule. Right? But as long as you're getting more than you're losing, then you're on the winning side of all of this, right? So what they do is they open their stomata at night. They don't lose as much water. They suck up the carbon dioxide. As the day starts, as the sun comes up and it starts to dry out, they close their stomata, just like everybody else. Deal with, you got the water thing taken care of because you're not dehydrating if your stomata are closed up. All right, good deal. Now, what do you do at that point? When the sunlight comes up, then you start releasing. This is all chemically happening here, folks. This is fixing and what did I coin it? Defixing. No, maybe not coined it. Defixing. You know, putting things on the molecules, taking them off, transporting. It's kind of like what NAD, FAD, NADP do with electrons. So this organic acid in oxalacetate and malate in the C3. Those were the organic acids you see on the left. But and I think, again, crassulacein is the organic acid here. Fixes carbon dioxide at night when the stomata are open. When the sun comes up and the stomata are closed, it releases carbon dioxide. And again, do the oxygen levels rise? Yeah, probably quicker here because it's all happening in the same place. But once again, as long as the CO2 levels are greater than the oxygen levels, and you hope that happens until the next night, or till the humidity isn't such a problem anymore, until it's not as dry anymore. You hope you can last that until then, and if you can, then you can live there. Mother Nature grand, man, she comes up with all these different ways of doing things. Now, let me ask you a question here just for a second. If I asked you to name an animal, no animals photosynthesize, no animals do the Calvin cycle, no animals do any of this stuff that's on the page. Let me say that. That's my little disclaimer right at the beginning, okay? But if we had to equate an animal in terms of how they what should I say, breathe, and how they breathe, which animal would be more like the sugar cane or the C4? Which animal would be more like the pineapple or the cam? Think about that. Now, I'm not talking about making food here, but I'm talking about an animal in general in terms of the way they breathe. And don't think chemically either. Don't get too deep. Which animal breathes more like the sugar cane, and which animal breathes more like the pineapple? And I'll add one more thing. And I'm thinking about an animal living in the ocean, living in the water. Maybe that'll help. Nobody has anything for me on that? I think that's a good question. So in terms of breathing, which animal on the left is able to get its oxygen through some sort of physical process versus some animal who actually breathes in oxygen? So if we're out in the ocean, what I'm basically asking you is who can hold their breath and live in the ocean, and who can't hold their breath and live in the ocean? Okay, fish don't have to hold their breath. Now, and dolphins don't have to hold their breath. You see what I mean? On the left-hand side, and maybe this helps you. Hopefully, don't give me a bunch of dolphins and fish on the exam, for goodness sakes. But what I'm saying on the left-hand side, what I'm trying to demonstrate overall is there's more than one way to accomplish something. If you live in the water, this is a very beyond the chemistry what's happening here. If you live in the water, one of the ways you can breathe, one of the ways you can respire, because fish still need oxygen. Everybody respires. Plants respire. You have to respire to survive. The only time you get exceptions or variants of respiration is when you start looking at bacteria and sometimes in animals. But everybody is going to respire. So you got to get oxygen out of the water some way, right? If you're not an anaerobic critter, even if you can anaerobically respire sometimes like we do, you can't do that forever. So the organism like the fish is able to extract the oxygen from the water through a chemical means. It's actually through diffusion. I don't need to go into it, but that's what's happening in the gills. The oxygen is diffusing into the fish's tissue. The animal on the right then, or the critter on the right, would be that holds its breath. All right, and some whales, how long can some whales hold their breath? How long do you think? All right, what's the longest amount of time? I'm going to look it up because I really don't know. I mean, I know kind of. All right, I got some numbers. How long can marine mammals hold their breath? How long can whales, dolphins? Bears can hold their breath for a while too, but just in general. Well, if you don't know, then look it up for me or look it up with me. I'm looking it up too. You might find out something that's kind of cool. You might leave the website up for later and just look at it if you're interested. If you're interested in zoology in any way, you know, this is a fascinating, because there's a lots of different, I don't know why I said it that way. There's lots of different things that animals do to be able to hold their breath as long as they can. Did I lose everybody? Come on, guys, do this with me. Do this with me. Wolves. What are wolves, Jessica? How long can you hold your breath? Yeah, that's probably about right. I don't think I could do it now. I think when I was younger. I don't know what the record is. Let's see. The record, 22 minutes. Holy cow. Anyway, all right, so you understand what I'm saying here? The dolphins and the whales and all, they can hold their breath up to like 20 minutes, I found. 20 to 30 minutes. That's amazing. And that's on a regular basis. That's why they can dive so deep. So they're doing kind of like cam planes. All right. Thank you for your contribution there, folks. Let's everybody stay awake. Almost done here. Okay, so that's all I'm going to say about photosynthesis. I can answer any questions you have, but if you don't mind, I would like to go to respiration, and I kind of want to just go through that. What time is it? 10:30. Perfect. I got 20 minutes to do it. Y'all ready? No, someone's. Oh, no, you don't. All right. I'm going to jump right ahead, though. Where am I? Here. That. Hold on. Let me see where I'm trying to go. I want to kind of try to make this mental connection if I can. There I am, right there. Okay. All right. You guys see the picture on the screen, right? Figure 9:15 in y'all's book. Okay, so if you saw a question on the exam, something along those lines, and I showed you this picture, first of all, with or without the labels. Now, I'm not gonna expect you, like I said in class, I'm not gonna expect you to memorize the names of these particles here. Okay, that's, if you know that they're helper particles that they help move the electron along, that would be good. But you don't need to know the names of them. They do have names, obviously. You can find about in their book. I do think it's important anyway that you understand that there are 3 pumps. Now I know they're named a little weird. That's because they're actually naming the proteins. But there are 3 pumps. There's this pump here. There's the one in the middle and the one on the right. Okay. So in an exam, I showed you all this sort of stuff, and I showed you that these electrons were pumping protons, and you can see quite clearly that's what's happening in the red arrows. And then, of course, the reason you do that. Right? The reason you do that is because you're trying to poke Mother Nature with a stick. You're trying to make her unhappy. She says, I don't like things out of balance, so those protons are going to flow back through here, and the living organism says, fine, do that. Every time you do that, I get to do this. And I use that ATP for building all kinds of different life on the planet. Now, if I gave you a picture of that and I said, which process in photosynthesis does something very similar to this occur? I'll type this question out for you. Which? I abbreviated photosynthesis. So which process of photosynthesis is very similar to the process that you see up here? During what part of photosynthesis do you get this going on? Not exactly necessarily, but the concept, the whole idea. Amber? I'm just going to go down the list. Colin, Gavin. How about you, Jessica, Kelsey, Kieran? Do you remember any part in the photosynthesis that we talked about in which something like this is happening? Marie, Sadie? Well, this here is oxidative phosphorylation, but do you remember somewhere during photosynthesis where it happened? Okay, chemiosmosis is what we're talking. Chemiosmosis is the process of the protons flowing through ATP synthase. What's happening right here, see how it's bracketed, is called chemiosmosis. Remember I said it doesn't have anything to do with water. It has to do with it should actually kind of be called chemidiffusion or just diffusion, because that's what's happening. The protons are diffusing across the membrane. But when they do, we engage a chemical process by which we turn it into ATP. All right, now again, back to the original question. Where during photosynthesis does that happen? We get a process very similar. Remember, okay, what I'm trying to get at here is what's the purpose of what you see on the picture here? Sometimes we lose sight of the forest for the trees. What's the purpose here? Why do living things do this? Yeah, Colin, there you go. And that's right, Marie, to respire, to use energy in food. But yeah, Colin, that's exactly where it happens. Photophosphorylation, okay? Let me, I got to switch between chapters, sorry. But let me show you why Colin said that, or at least why I hope Colin said that. All right, again, what's the big picture here? The big picture is some process is causing a disequilibrium. It's like a detective. Something is going on here. There are more protons on one side of the membrane than the other, the way that the arrows are showing. And the reason why that's happening is whatever this process that's happening, in this case, it's this whole thing with 3 pumps, you're creating a disequilibrium. So if you can make this disequilibrium, then you can use it. Living things can use it chemically to their advantage because they can use it to drive this reaction. And once again, that's the reason why we're doing respiration, folks. We do it to make ATPs. That's the reason. Because you need those. Those are the things that go out throughout your body and say, oh, got a little cut here. Here's the energy you need. Blah, 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 fix you all up again. Right? That's your maintenance energy that you need. Remember, that was the purpose that occurred during respiration. All right, now let me go over to photosynthesis and show you where I'm hoping Colin was, or what Colin was thinking about. Why isn't it scrolling on? Right. There you go. Right there. See? Same thing. Same thing. Same thing. Now, compare and contrast, right? Same thing. Overall idea. Overall, like the whole purpose behind why you're doing this non-cyclic photophosphorylation. The name tells you, not the non-cyclic part really, but the photophosphorylation. You're putting a phosphate onto something. So when you're studying these names and you're learning these names, think about the names. Think about what it means, as we've talked about in class. Think about what it means to phosphorylize something. That's okay. Some of you can write on the screen. The other you can tell me if you can. Where in this whole picture is phosphorylation happening? Point to it, circle it. Tell me if you like. I don't care. Where in this picture is phosphorylation happening? Okay, first answer gets a point on their next exam. I'm going to write this down. First one that gives me the answer, first one that points to it, says it, gets a point on their next exam. Nobody? Nobody wants a point? I'm not going to raise it anymore. They could give you 89, 90. Yeah, where? Where does that happen? Kieran, where does it happen? Tell me where. You at this point, where in the picture? Well, I'm certainly not going to detract a point if you guess and get it wrong. So if you are listening, you, by all means, I guess. There you go. Marie Congratulations. Add a point to your exam. I just wrote it down. Hold on. Just drop my pen. All right. So this is the process. And I'll tell you what, another point, where you can't join in this time, but another point, if you can tell me why it's called photo. Why is it called photo? What's driving this process? Colin, good man. All right, good deal. All right, you see, we are transferring energy because who answered? Marie. When Marie told me where ATP synthase was happening, okay, that was obviously an endergonic reaction. If you're dephosphorylating something, it's not endergonic, but if you're phosphorylating, it is. And Colin came in and understand that energy to do that is coming from the light of the sun. The light of the sun. All right, now contrast that in respiration. Back to the other chapter. I don't have time to switch back to it. Where does the energy come from to make this imbalance of protons? To pump. 1 point. 1 point on the exam. Colin and Marie, you're exempt from this. Yes. No. Respiration, not photosynthesis. Where does the energy come from to make those protons be pumped across the membrane? No. Okay, think back to the whole process of photosynthesis. No. That's what you're going to make out of this process, but where do you get the energy to make that stuff? Okay, think about the whole result of photosynthesis. Why do things photosynthesize? Food. Thank you, Sadie. You get the energy from food, folks. So if you were looking at this picture and I said, all right, this is photosynthesis, notice what's happening. Tell me how this is like respiration. You'd say, okay, well, we're pumping protons. Ah, there we go. Why we pump? Oh, look at that. It's even got the same name, ATP synthase. Again, I'm not going to switch back to the other picture. So if you got it open, take a look at it. Look in your book. It's the same doggone particle. So Mother Nature says, I'm going to make this particle. If you can figure out some way to make this disequilibrium of protons, getting ready to use the real pen. If you can figure out some way to do this there, living thingamabob, then I'm going to let you actually do this phosphorylation process. And guess what? Then you can make big trees and fish and dolphins and bacteria and fungi and all kinds of living things. If you could take the energy from the sun, which is what you're doing, and you can turn it into complexity of your body, of a living organism, that's what you're doing in the long term, okay? You're getting the energy from food, because you can't photosynthesize. Plants get the energy from food, they just happen to make it. But originally they get energy from the sun. And the way you get the energy is simply by setting up this disequilibrium. It's all about the protons. It's all about the protons in photosynthesis, okay? It's all about the protons in respiration. You're making ATPs in respiration, but remember, you make a lot of them at the end. A lot of them during that oxidative phosphorylation. Okay, if I asked you to talk about the differences, all right, the differences. Well, obviously, if you're getting the energy from food in respiration, right, you don't need to get the energy from sunlight in photosynthesis, right? You got to have energy to drive this reaction. Remember, the laws of thermodynamics are saying that you're building up a complex molecule. Okay, now back in respiration, that complex molecule happens to be ATP. It happens to be ATP here too, but that's not the end of it, right? The complex molecule that you're making is food. What you're doing, and that's in the dark reactions, that's over here, remember? Carbon dioxide is being turned into food. This is Arbeiten Zusammen again, folks. This is the stuff that's happening in the light reactions, the products that are being made, used to do another set of building up, endergonic, energy-needing reactions. You get it? See how you got to start thinking about this stuff in context. It's really going to help your understanding. It's going to help you on the exam too. All right, you're very likely, I don't remember which exam version I want to use, but you're very likely to see a question where you understand the differences and the similarities between photosynthesis and respiration. I love compare and contrast questions. Compare and contrast questions are the best way for me to understand how well you can tell me what the differences and the similarities are. You know, what I'm trying to do here is to help you understand how to learn, how to find, and I know many of you do, don't get me wrong, but I'm just saying to those of you that might need some help with that, I'm trying to help you build connections. Don't just memorize the facts, memorize what's going on, okay? So like with this, you know how I would study this? I draw it. I get myself a blank piece of paper and I draw out the thylakoids, and I would draw the photosystems, and I would draw all this stuff that's happening here. If I had to actually look at it and sort of memorize the beginning, I would. Even if you have to memorize it, that's fine, as long as you can get it drawn down. So you could do that on your scratch paper during the exam, or you have the picture right in front of you. And since you've memorized where all the parts are, you can now easily describe the function. Okay. So, I mean, that's how you study it. Draw the membrane. Okay, membrane, then I got photosystem 2, and then there's the cytochrome complex, and there's a couple of helper particles. Photosystem 1, there's NADP plus reductase, there's ATP synthase. Boom, boom, boom. Okay, what do? Protons flow through. Okay. All right, let's go to the other cycle that occurs during this process too. Remember, there was the Calvin-Benson cycle, the dark reactions, the Calvin cycle. Oh, crud. Hold on, I do have to change slides. Hold on, let me go to the other ones. Back to respiration. Hold on one second, folks. I'll get it for you. Okay. All right, here we are back in respiration. All right, so this is another great place that you might think about comparing and contrasting, because it's not important to me anyway. It's not. Hey, notice there's a couple of molecules in here that we talked about in the C4 plants, right? Malate and oxaloacetate. You'll find out that, and there's, I don't know exactly what that means about how closely related these kinds of things are, but you'll find that Mother Nature, in spite of her complexity, have found some really tried and true ways of doing things. So apparently when it comes to this whole chemical reaction and process, malate and oxaloacetate interacting with each other is something that's done rather commonly. Okay, so Calvin-Benson cycle, dark reactions, Calvin cycle, that's the making of food and photosynthesis. Don't confuse it with this one. This one is known as the citric acid cycle. Now, the reason why it's citric acid, if you're going, what does it have to do with oranges? Well, oranges have citric acid, and citrate is citric acid, or close to citric acid. It may not be exactly citric acid. Okay, so let's go with some similarities and comparisons here. Let's start right off the bat. So for 1 point, someone tell me give me one similarity between the citric acid cycle and the Calvin-Benson, or the Calvin cycle. Again, those you've already answered, you're out of the loop. So it's kind of like inhibition and enzymes. So give me one similarity. It can be a similarity in something you recognize in one of the parts, or it can be a similarity in a process you recognize. I should play the Jeopardy theme. I should somehow get that. So we've got the citric acid cycle, right? Yeah. And I'm asking you to compare that to the Calvin-Benson cycle. So what's one of the similarities between these two processes that we're talking about here? Amber, Jessica, Chelsea, Kirian, see Shayla. Any y'all have any idea? Well, either you've lost connection or you aren't listening to me, but you missed your chance at a point. Anybody of the three that have already answered? Anybody? Okay, they're a cycle. There you go. I like it. Yes, they're a cycle. Nice. There you go, Carrie. Nice job. Marie. Same thing. Yes. It makes ATP. Oh, wait. Now say, look at your answer again. And I can't go back to the Calvin-Benson cycle right away because I don't want to switch over. But does the Calvin-Benson cycle make ATP? See if you can look back in your notes or in your book. Does that would be a good contrast? And like I said, compare and contrast what's going on with ATP here. Yeah, no, it doesn't make ATP. Instead of making ATP, what's it do? So Sadie, instead of making ATP in the Calvin cycle, what's happening? Not making ATP. Yeah. It uses ATP. That's right. It uses ATP in the Calvin cycle, okay? Here it's making ATP down the lower left. Now, the reason why I'm kind of harping on that difference is that explains a lot about understanding the exergonic and the endergonic. Remember, the citric acid cycle respiration, part of that, right, is exergonic. It's releasing energy, all right, and it's using that energy to make ATP. Now, the other one, photosynthesis, is endergonic because you're using energy to build something up. Don't get confused back here of the ATP at the bottom. Remember, this is the ATP in respiration that's being made for everyday cell use. The ATP that's being made in photosynthesis has that special job. Now, I'm not saying that the ATPs are slated. You shall become one that works in the Calvin cycle. You shall become one that helps the plants turn its leaves towards the sun. I'm not saying that. I'm saying that some of the ATP made during photosynthesis, during the light reaction, is going to be used in the Calvin cycle, okay? So it's the difference between using and making ATP. It's the difference between exergonic and an endergonic reaction, right? Okay, now finally, one more point. Can you tell me, no. Oh, good question, no, Sadie, good. Look in the lower left. You're creating ATP. Be careful to follow the arrows. And understand it's a little complex, but remember, you make GTP, which is kind of like ATP. GTP then makes ATP. So even though it's one step in between, you're still making ATPs in this one. This is respiration. In photosynthesis, you're using ATPs. Okay, good. All right, so I'm sure I don't have time to go over all of these kinds of things that I want you to look at, but I'm hoping that you're getting an idea of what I want you to look at, of the importance. Let me pull up one more thing before I leave. What time is it? 10:49. Okay, I got one more. I want to make sure that you go back. Of course, all this is important. Don't get me wrong, but I want to make sure that you go back and you understand glycolysis again. That very first step. Okay, everybody does glycolysis. Glycolysis happens in the cytoplasm. Okay, this is all respiration again. The mitochondria, all that other stuff, that's where oxidative phosphorylation, that thing with the pumps, that's where that happens. So when you're respiring, glycolysis happens in the cytoplasm. All cells got cytoplasm. Bacterial cells, archaean cells, all prokaryotes, eukaryotes, everybody does glycolysis. Glycolysis works. Some organisms just use glycolysis. Now, in glycolysis, there is an investment phase followed by payoff. It's all about the ATPs. You got to put 2 in, you get 4 out. Okay, so the total yield of ATPs is 4. The net yield is 2. But as long as you're making more than you're using, as long as you get 4, and you know we need to use 2, you're okay. And remember, the reason why you get 4 down below is because very ingeniously, right at the very end, that enzyme splits the molecule in half. Okay. All right, that's class. Any questions?